Because I said what was on my mind. I made a lot of enemies. I made a lot of enemies.
Welcome, everybody, to another episode of In Defense of Virology. My name is Bryce Nichols. I'm a professor of genetics at Rutgers University. And as always, I am joined by Simon Wayne Hobson. Simon, how are you doing today?
Hello, Bryce. Lovely to see you. Today, we're going to have John Lennon playing on a white Steinway piano. This is a photo goodness me, I hate to tell you how many years it goes back, but it's nearly 60. And I'm staying with my son, so I thought this would be a good backdrop to our conversation.
Today, Simon, let's talk about two things. The first thing is it was recently revealed that you gave a talk at the NIH. And I guess I had maybe seen evidence of said talk on Reddit. Somebody posted on Reddit that a quote-unquote right-wing podcaster had been invited to speak at the NIH. And it turned out that right-wing podcaster was you.
It's universally known that I'm a right-wing podcaster. I mean, everyone knows that. And I come from a family of artists, and therefore it's obvious I would be right-wing.
Well, we'll let the audience judge whether or not you're right-wing.
Yes.
Yeah, we can't say whether you are or not.
We will not jump to conclusions.
But we can say that you did speak. You did give a talk at the NIH, which was revealed recently, leaked somehow to the press.
Yes.
And so do you want to say something about the circumstances around that?
Yes. Well, I mean, I can't speak to the leak. I know nothing of it, but I was invited by the people in the NIAID to speak there. And the title was Dangerous Gain of Function or Gain of Function? That is the question. And readers and listeners will know that many virologists have played on what is dangerous and what is not, in a hope of fudging so that we just keep everything going on, basically saying it's so hard to differentiate between the two, let's keep the existing criteria. And the NIH were interested in, well, what are your thoughts on this? How can we separate the two? And I went into it in I hope in the same way that I speak to you, try to explain my opinion. I don't want to ram anything down anyone's throat. And basically said, if you're making the equivalent of novel biological weapons out of animal viruses, which might harm maybe just one person, but what if they had the potential to harm, 50 million with the death rate of maybe 3 million? That is not on the cards for me. And therefore, I try to give a number of criteria, the first one being do no harm. The other line I use is don't make the world a more dangerous place. And just remember what we virologists are supposed to be doing. And I think I said something like, we're here to sort of help suffering and disease and people in pain. And yes, we learn lots of intricate details about viruses and things that, incomprehensible to most people, including most virologists. Sometimes it's so esoteric. And but we are the big picture is we try to make diagnostics, vaccines, drugs, and for example, drugs is you can come up with an interesting target and try and make a drug, and academic labs can do this, as can industry, and so on. But that's our ultimate thing, is to alleviate and fight pain, suffering, not just in France and US, but everywhere. And I gave a number of criteria and a number of ideas. And I think one of the things I said is that you need to get a change in the mentality of the scientists, because they know the science better than anyone else. We're talking about really specialist knowledge. And therefore, there's not too many people who know that. So the people with this specialist knowledge, you have to be sure that they will use that specialist knowledge carefully and get them to sort of first say, well, do you think this has got any element of dangerous gain of function? Don't think of the NIH and who are going to be policing it or reading it or checking it. The NIH shouldn't play the policeman. It's the scientist who has to police themselves, and then they will come in afterwards. But if it's a game of, are you going to find out if I've submitted a dangerous experiment or not, and I can maybe hide it or disguise it in esoteric facts, I think that's not fair. So they have to come in. We need a change of mentality. And you and I have often said that we need a Hippocratic oath for STEM scientists, so that's science, technology, engineering, and mathematics. At the end of their thesis fiber, we need something. They need the people to have to stand up and give their opinion first. The NIH can then disagree with them, but they have to tell us, and they have to be honest. And I think that's the first step. And we spoke about many other things, and there was a question answer session, and we spoke for nearly an hour and a half.
How was your talk generally received?
I've got some feedback and people saying that it was appreciated by quite a number of people. I don't expect to have everyone agree with me. That would be bad if everyone agreed with me. But so if it was useful, fine, terrific. Some people said it was, so and reached out to me. So if I could help a few people, that's what I'm around to do.
There were many people that were happy to learn that your voice was included at the NIH, that you spoke in front of the NIAID. I think was welcome news to a lot of people.
Yes, it's the first time I've ever spoken to an American organization about gain of function since I've been involved in 2012.
And so this also comes alongside, Matt Ridley's talk on COVID origins that just occurred, a
Yes,
few weeks
indeed.
ago.
That's right. That's right.
So these are positive steps.
Yes, it would seem, yes. And let's see how they can, yeah. Because they do have a problem in as much that most of the virologists are against it. So they have to make a serious job if they're going to do this, because we can't just move out a whole army of virologists and recruit them, because you'd lose so much special knowledge. You would do such great harm to virology. So you've got to change the mentality and tell the scientists that, well, some things you can't do.
I'm suspicious that only people with British accents are allowed to speak at NIH on this subject. I'm wondering if that's
Well,
something that maybe there's something to that.
I would not. You've got 2 data points. That's a bit thin. So maybe 2 points isn't quite enough. But if they invite a third Brit, then you have a good hypothesis.
Okay, fair enough. And
Okay.
right. So we'll see. We'll see who the next one to speak there is, and if they are British, then I will
Oh,
be right, and
uh,
you will be.
let's move on. Let's move
All
on.
right, here we go. Second subject is the recent Cell paper titled Dynamics of Natural Selection Preceding Human Viral Epidemics and Pandemics, published in Cell and covered in the New York Times in an article by Carl Zimmer called Scientists Get a Glimpse on How New Pandemics Are Made, as well as other news organizations. The Telegraph also covered it. And the title of the Telegraph article is A Nail in the Coffin for the Lab Leak Theory. Tell us a little bit about this paper in Cell.
This is from the group that has a long track record of arguing from the natural zoonosis point of view. And every 6 months they come up with a new analysis showing why this virus is natural and not an issue to a lab leak. Here, they try to address that problem more directly. They say, well, we're going to be looking at the evolution of SARS-2, Marburg, Ebola, H5N1 flu, and a number of other viruses. And then we're going to, later on, they look at the evolution of 6 viruses in tissue culture, that is, that have been passaged sequentially in the lab. Passage is a virological term. Basically, you grow the virus, it consumes all the cells. You have to then take an aliquot of that and grow it on fresh cells. And you do this process, and it can be, some people have been doing it for 60 to 300 times it happens. So they have these data sets that they pick from the literature. And they say something in very early on in the abstract. They say, we conclude that extensive pre-zoonotic adaptation is not necessary for human-to-human transmission. Now, the whole thing about evolution is that when the environment changes, the host, which is, say, maybe a mammal, a plant, or in our case, viruses, there will be genomes within those virus sample, which will allow you to adapt to the new environment. And that one will be selected because the environment has changed. You can only adapt to humans once in humans. So the idea that, say, a bat coronavirus is perfect for humans is, well, firstly, you have to prove it, and they don't. But the slightest change will alter the virus. I can give you a case for HIV-1, HIV-2, very interesting case. So it goes back to my HIV days. HIV-2 just was the epidemic here centered on Senegal and the Gambia. And it wasn't as aggressive as human HIV-1. And when you put this virus into culture, there was a very particular singular mutation that occurred in one of the genes. And people observed it, and it's been known for ages that when you culture a virus phrased from blood, you will select out a variant. And my lab showed this, was the first to show this for HIV back in 1989. But you can look at bacteria and parasites, and as soon as you culture them, their genomes start to change. So the interesting thing about HIV-2 is you then take this virus with this singular mutation, put it back into the rhesus macaque, because, of course, you can't put it into the humans, but people found pretty quickly that the virus grew in rhesus macaques. And what happens? This mutation goes back to the original amino acid. So the virus is tissue cultured. It changes to adapt to that new environment. You put it back, and then you put it back into the macaque, and the thing reverts. It goes back to the original. So it shows you that when you culture viruses, they don't recapitulate the disease in humans. I remember I first started virology back in 1980. I was working on hepatitis B, and quickly I learned that the specialists who were studying hepatitis B in chimps were saying that HBV, hepatitis B in chimps, doesn't fully recapitulate human disease. So the chimps are roughly 99% identical to us, and yet the disease spectrum is different. So when they say that there's no adaptation for human-to-human transmission, I don't believe for a second. So it starts out with a statement which I find extremely different and flies in the face of what most people have been doing. So I get the impression they're mixing up terms, and of course we know why. They want desperately to show, but we'll come to this. So I don't think they had a clear idea in mind. And therefore, just on that alone, I would, if I had been a referee, I would have said, no, I don't, this is a false hypothesis you're building. It's a straw man. I don't know why you're doing it.
This is an example. This paper appears to be an example where they're simply trying to find something that they can publish that points to a natural origin of SARS-CoV-2 and sort of working backwards from there. The essay that's going to come out on Biosafety Now Substack goes through the flawed logic of the paper, but just starting at the first principle here, is this the way science should be done, frankly? Maybe maybe the question to that end is, say they got a different result. Say that this methodology that they developed pointed to a lab origin of SARS-CoV-2. Would they have published that, is perhaps my question. I don't think they would have.
I don't want to go down. I don't want to get
Well,
I
but
don't want to get inside their heads. It's my own head is complicated enough, and
well,
and
but
you're
why do but if you're starting backward, if the point of this paper is to reach a conclusion that points to a natural origin, is that the way science should be done?
no, you too. I mean, we are humans with our passions and our faults, and you have. It's not possible to be completely neutral. I mean, but you have to be aware that your idea has to be ultimately supported by data. And if it doesn't, then change your idea fast before someone will change it for you. Because to be shown to be wrong as a scientist is something that's very painful and it hurts. So better, so if you've got something, if you're making a mistake, you're going down a path that you didn't quite perceive that it was the wrong path, then change. And if you show that you were wrong and you said, no, but I then do a sidetrack and I get it right, people will say, terrific, you had a flawed hypothesis you realized, and actually it told you something that you hadn't perceived, and you move on. Everyone will say, that is how science is done, trial and error. But when you continue to for too long, to push a hypothesis that's way before its best before date, let's be clear and use a phrase that I hope your readers will understand. This is Orwellian science. This is 2 + 2 = 5. Okay? It's not 2 + 2 = 4.
But getting it published in Cell creates this illusion, I guess, that this is real science and that this is meaningful and extremely high impact and all kinds of problems, right?
Yes, and therefore it was in review process for 13 months, which is rather long. The essay ends, I'm sorry to go to the end, but the essay ends with this remark. Artemis 2 didn't go to the moon and back based on Cell papers like this. That, of course, is my opinion. Other people can disagree with me, but it's my opinion. It's not good enough for Cell. And I don't know why they published it, but there we are. Now, but we'll get on into this. So they then go to look at how pandemic flu, Ebola, Marburg, and they develop some criteria. They try and describe the evolution of a virus in vivo in a single number, which, I mean, for someone who's been working on viruses, the idea that you can resume a virus in a single number is just doesn't make sense. The evolution of viruses is multigenic, and therefore you could have a plus cancelling out a negative, and then that information of those two genes is lost. A positive regulator and an amplificator are like, the brake on the accelerator of a car. If you put that all into one number, you might forget the fact that a car has something that goes, you push the gas and then you put your foot on the brake. You lose that concept, as though this thing somehow does it automatically. So I found that unsatisfying. And then they do all this, and then they do this by aligning the genetic sequences of a number of viruses. So if like SARS, they'll align 20 or 30. And of course, there are these little deletions in genetic sequences. They can be as small as 3 bases, and they can sometimes be as large as 1,000 bases, if not more. But when you've got genomes with these little insertions and deletions, you can't look at what we call single point mutations. So, for example, if I had sickle cell anemia, I would have a single mutation in my beta globin gene. So that they would score. But if I had an insertion or deletion just out of that gene, that would be lost. So they align all these things, and that's what they analyze. Now, as listeners will know, in SARS-CoV-2, there's something called the Furin cleavage site, which we know from experimentation Subsequently, when proximal origins came out, this was all speculation. But there's been lots of experiments now, and we know that Furin cleavage site is hugely important to the virus. Now, that Furin cleavage site is deleted from this analysis. It's not considered because it's not present in all the other SARS bat-like coronaviruses. So the analysis is missing some hugely important pieces of information that we know to be important. And therefore, you could say that what they say, basically, that we don't cite evidence for. They're saying, and I'll get the exact term, so I'm not going to abuse language. We conclude that extensive pre-zoonotic adaptation is not necessary for human-to-human transmission. Now, arguing a negative in science, as you know, is incredibly difficult. So let's go to the Sherlock Holmes, wonderful Sherlock Holmes example. Why did the dog not bark? Okay, what is it? The Hound of the Baskervilles, isn't that the one? So the most plausible explanation is that it knew the intruder, and that's what Sherlock Holmes deduced. But in our modern world, we know that that dog could have been drugged. What if that dog had laryngitis and couldn't bark? Not unlikely, but whatever. And what if it was, and this is in my essay, I couldn't resist. I said, what if it was too absorbed with canine social media and was simply distracted? And therefore, was so interested in, they just didn't pay attention. Or was the dog deaf? Improbable, low probability. But when you argue a negative, it's very difficult. And in science, sometimes the list, is very long. So it's very hard to prove a negative. So they tried to do something that is intrinsically hugely difficult, and fraught with difficulties that I won't say a rookie scientist would necessarily know, but once you've got, say, 10 years of science after your thesis, you know that proving a negative is very, very difficult, and you should be careful. And they go on trying and want to prove a negative. Now, do they prove a negative? No, they don't prove anything.
Pre-adaptation, if you would also comment on that, like, is pre-adaptation a thing? That's also something that you talk about in your essay.
And actually, this is rather interesting because so many people said it's amazing how this virus was so well adapted to humans. Now, we have a paper from, and I mentioned this, a paper of Peter Daszak, going back a number of years, and they showed that there are a large number of spillover infections to humans from bats, which they do this by serological methods, okay? And they do this, and they show that something like they estimate that 66,280 people are infected with SARS-like coronaviruses from bats annually in Southeast Asia. Now, the whole of the flu and the H5N1 gain-of-function debate was around spillover events of bird flu into humans, some of which were deadly, and they could kill maybe 30, 40, 50, 60% of people. And Kawaoka and Karushi said, well, what if these viruses could then transmit? So by what we know is that these SARS coronaviruses, apart from SARS-1, which burnt itself out, and SARS-2, the vast majority of these things are spillover infection, and they don't go any further. They grow inside the human body because they did this by looking for antibodies, and it's just something your listeners might not know, but you can't make antibodies if the virus doesn't grow. It has to grow to a certain level to make the antibody, okay? And so therefore, they grow in humans, they make antibodies, and the immune system eliminate, but they don't transmit. So this means that these viruses infect humans, and therefore to infect, that's pretty good adaptation. adaptation, but it's not enough for human transmission, for human-to-human transmission. So this says that, these back coronavirus haven't got it all. Yes, some, and if we take H5N1, which we know, they grow so well that they kill 60% of people, but they don't transmit. And since 1997, we haven't seen a human or very many cases of H5N1 human-to-human transmission. So these viruses can't do it. They're not pre-adapted. They're pretty good, but they don't have it all. So the whole pre-adaptation thing doesn't make any sense. Anyway, so they compare this to evolution in viruses in tissue culture, and then they sort of fudge like this and say, you see, because we didn't see this, therefore it proves that this virus did not have any time in tissue culture. And they say they find this for H5N1. Fine, we know H5N1 was cultured, but H5N1 doesn't mean to say that the SARS coronavirus it behaves in the same way. So they make this comparison. At best, they can make a correlation, but correlation is anything but proof. And maybe a paper with a correlation you could find published in a small journal in Virology, but not in the top of the top like Cell. I wouldn't say it's a correlation, but let's say let's concede that it's a correlation, but so what? That doesn't prove anything. The correlation doesn't prove anything.
My opinion is that this paper was designed to simply provide evidence that COVID-19 emerged from nature, not from a lab, which I think means that it's flawed from the get-go. And then the other piece here is that they're coming up with a method that they are claiming can allow people maybe to distinguish between things emerging from nature and from a lab. And the goal then might be to apply that method in the future. So I would then say that if this paper is published in Cell, that presents a problem, because that will be viewed as the scientific community as like, oh, well, this is a very solid method published in a top journal that we can apply to future outbreaks to see if it's natural or lab derived. But if this method is like junk and meaningless, then we're just doing, I don't know, but doing more harm to the scientific enterprise. And
as I said, Cell papers when I was young, everyone dreamed of getting a paper in Cell, and I've had papers in Cell, but that they publish something this sort, they should not have. It doesn't make any sense, and therefore it smells as though, or feels as though they're complicit. But that I'm not making an accusation because I just don't know, but it does make sense. All I can say that this gentleman behind me, he would say, "Help! I need somebody." Right. So we need somebody, because the journal, sometimes the journals are going mad when it's COVID origins. Reason seems to be getting pushed aside, and agendas are being pushed instead of reason, and that is never good.
And, the idea here is, and I will say it, that Cell, because they hung their hat on this being a natural origin and have published other papers on this, that they're simply doubling down and they will accept things that are aligned with that because it would perhaps be in the interest. But there might be incentives to do that. But
Since when has a journal the right to publish anything has a hidden agenda? They're not supposed to have any agenda whatsoever. They are supposed to consult the knowledgeable people and get an opinion and then publish it independently. But I will point out, as we've seen for dangerous gain of function and COVID origins, that the whole of the virological community are in favor of continuing this dangerous gain of function. They say it's all zoonosis when they do not have the data. They don't have the data. They say it's zoonotic, as we've seen in some previous essays of mine. And when you look at the data, it's more an opinion than anything else.
last episode, we talked about peer review, the problems of peer review, and I mentioned if we could get rid of the journal system, that would go a long way to helping solve some of these issues. And this, to me, is an example of that, because putting a paper of this low quality in a high prestige journal like Cell creates problems in the future. And it's simply just not good science. And platforming it in a top journal is like amplifies the problem.
And it's amplified by Carl Zimmer in the New York Times, which is, if you want a big foghorn for science, you read the New York Times if you're American. And so he picks this up and he goes with it, and he makes the same mistake that we looked at in our last talk and in a couple of papers. They say most of the peer-reviewed scientific evidence supports this thesis. Well, as every single virologist, apart from me and maybe one or two people in private, say it's natural, then obvious, because all the people who are going to say, well, have you discussed the lab leak? They will say, where's your data? Well, question. When people withholding data, and this is very clearly said in the Sago report that I published last week, the Sago report says, well, we didn't have data on. We asked for the Chinese, we asked from the intelligence service, they provided us with nothing. So there's data that's lacking. There's data being withheld, and yet you give an opinion. Now, I will come to my little beef again. You've heard this story that RETG and the SARS coronavirus are 96.4 identical. Okay, this is the best we have. Well, we should have a genome that's 99% identical, if not 99.8. That would satisfy us. But we only have 96.4, and this is RETG. And we're 6 years down the line, 25 million people dead, and we can't do better than less than 100 bat virus genomes. And the closest is 96.4. Let's forget your remarks about RETG 13 that some people don't like. The genetic distance between humans and chimp are 99%. And we all agree that humans come from chimps, but there are some big differences between humans and chimps. One is that we have one chromosome less, which is due to the fusion of two chromosome 2-like chromosomes in the chimp, and we have one chromosome 2. They have the two. All that information is in 2, so they have one extra chromosome that we don't. And but suppose now for lack of sampling, we did not have the chimp, and we looked. Our closest relative was 96% identical. That closest relative is the gibbon. Now, I think you just have to look at the gibbon and humans and see there's a huge number of differences between the two. And to sort of say, well, we've got RETG, and wherefore it's just a matter of filling in the dots, and you'll get to SARS-CoV-2. The chimp and human are different. I told you that hepatitis B disease in humans and chimps is subtly different. And you get to the gibbon, which is very different. It's beyond orangutan, and there's a whole lot of evolution. And you pretend to say that you can honestly say, yes, case closed, it's a natural infection, when you've got all that evolution, which we know, because between gibbons and us, we've got what? We've got the orangutan, then we've got the chimp and the bonobo, and then we have humans. And the macaque is further behind. Okay? So we know there's a huge amount missing, and they say, it's natural. It's just a joke. It's just a joke. These are agendas that do not stand up to scrutiny. And we see it again with this paper.
It's an indictment of the field of virology, frankly, Simon. I mean, that's the problem. Like, the field of virology is failing on the science piece of this. That's it. And like you said, this is just another one of the papers that has been put out that doesn't help. And in fact, perhaps, it's rinse and repeat. You do a high impact paper and a New York Times article accompanying it. And it tends to happen around the same time each year.
Yes, and it goes a little bit beyond because the story was picked up by someone who writes for the Telegraph, and he takes some cheap shots at the NIH director. This is a medical historian, so he has a PhD in history. He doesn't have a PhD in life sciences, and he has a shot at Matt Ridley, who is a science journalist everyone knows. But Matt actually has a PhD in zoology and basically writes science books. So he knows probably, if you just compare him to this gentleman, he's better prepared than this writer who is a professor of history in the UK. And yet he cares to say that Matt Ridley is a science journalist, not a virologist, and a leading proponent of the lab leak theory. So you see that he's not a virologist. And therefore, this is meant to discredit him. The writer of those lines is not a virologist, but he doesn't see that his own writings pertain to himself. And therefore, why should I believe a man who is not a virologist talking about virology? I would say you can disagree with Matt Ridley, that's your right, that's almost like a First Amendment right. But Matt Ridley has a PhD in virology, and he's been speaking about biology and writing about biology for ages, and still is publishing books, some of which are bestsellers. I spoke to a Dutch journalist just yesterday, and he was asking me, why is it that so many, or nearly all the virologists say we should be doing dangerous gain of function, and that the COVID-2 virus is a natural zoonosis, and you just say, we just don't have the data. But he said the vast majority I speak to, almost everyone I speak to, says it's natural. They don't have the data. And yet they say this. This is, people are afraid of admitting that science might have made a mistake. And in the case of dangerous gain of function, that mistake is making the equivalent of a biological weapon. Instead of being made by military people who have rules unto their own and secrecy, if the military made a biological weapon, the last thing they would do is publish the recipe in Nature or Science, because then an adversary could take that recipe and do it themselves. So the civilians, and with their complicity of Nature and Science, they publish this, and it's the equivalent of biological weapon. I mean, it's just it is madness. And people are saying X and Y, and that as we come down in defense of virology, I might be the only virologist, but I'm trying to sort of say, virology is too important to have it mauled and messed around, and for people to lose trust in virology. It's a beautiful science. It's fascinating. It's complex. It's interesting. It's subtle. It's important. It's an extremely interesting and rich subject, and very fulfilling because you can actually do some good.
The what you just said, though, is extremely disturbing, Simon. If the entire, most of this field of virology is in denial of the potential misuse of the science, then frankly, they're going to take out all of science, not just virology. That was my concern with Richard and Justin Kinney when we started Biosafety Now, was that the virology community is threatening all of science, not just virology.
Yes.
And that's, yeah. So in defense of virology, in many ways, is actually in defense of science. And I don't, I think it's a good step. I think it's a good step that you're speaking at the NIH. I think it's a good step that Matt Ridley is speaking at the NIH. But until the virology community at large is able to perform science rather than obfuscation, this is
Yes,
deeply,
and
deeply disturbing.
Therefore, there'll be calls to cut back HHS budgets to NIH and NIAID, and in that, there might be less work for cancer. Maybe we can cure one cancer, 60% of cancers, but your listeners will know pancreatic, ovarian, and some extremely aggressive cancers, which we still can't, or people relapse after some of these immunotherapies pretty fast. Alzheimer's, all these metabolic diseases of a certain age, Parkinson's, There's a lot to do. And yes, all these other subjects, I hope to be a little bit more modest and say in defense of science, because I'm a virologist and I don't want to be pretentious. Virology.
Well, no, it's just virology. You can't.
You're absolutely right. You're absolutely right. If we talk about trust, it's trust in science. It's not just we trust in virology. Everything will get hit by ricochet, and this is bad. And this is bad. And I can't get people to see this. And if I may say, this goes back. It goes back to a number of things. We've created a sort of an approach with the human genome, everything that science can deliver, and we can do everything. And of course, listeners will know that we don't have an efficient tuberculosis vaccine. Well, that's 148 years since that bacterium was isolated, and we don't have that. Only recently we have a chance of getting hold on malaria through vaccination. So there's a lot of things we need to do. And so we've got this sort of can-do, leave it to the experts type of machismo that's developed. And for the dangerous gain of function and COVID origins, I think this editorial in the Washington Post in December of 2011 from Dr. Fauci and Collins and their colleague Gary Nabel, and in the Washington Post saying that this was a flu risk worth taking. The Kawoka and Fauci experiments was good science. This I've banged away at this opinion piece. I've written an essay on it called Chilled Virology. This sent a chill through the whole of virology. Who would get up and say these two gentlemen, Doctors Fauci and Collins, were wrong? They wouldn't. So basically, no one would speak about dangerous gain of function because two administrators had said this. But I'm sorry, science knowledge is not for administrators to create. It's for scientists, people I say at the coalface, people on the bench. They're doing the work. Then maybe we can generate a consensus. I mean, think of your field. Some there are areas of bacterial genetics that take, 3, 4, 5, 10 years to sort out sometimes. And as I said, we don't have an efficient tuberculosis vaccine. So some things are hard, and we can't have science administrators just suddenly saying, let's go to Europe. We can't have administrators in France, Germany, Switzerland, and elsewhere, Japan, Australia, saying this is what science is. We've got to let the scientists do it. And this thing stopped any criticism. And with COVID origins, well, you know this, your readers know because they're US-based, there were many policies that were maintained when they should have been modified because there was data around and people said, no, we've made a decision, we've got to stick with it. And science is about changing and modifying as things go along, as data comes in. So that I think that op-ed in the Washington Post is an opinion. Therefore, you can say it was their Fifth Amendment right, their First Amendment right to write it. It had a very damaging effect, and unfortunately, I'm down. People can check. I call it the equivalent of a papal bull. The Pope has decided this. That's it. That's not the way science is done.
It's been difficult to unwind that, and we're still trying to unwind that to this day.
Yes.
In the field of virology.
And there's resistance.
This idea about administrators or, defining what should be done scientifically or what science is, to this point, I do actually think that it might be of use for university administrators, to consider science and the research that occurs at their universities can, unlike other aspects of the university ecosystem, cause tremendous harm to the public. And
Yes.
that makes it an unusual category of stuff going on in a university.
Yes.
So perhaps in that regard, universities may want to consider what what their goal is and what is science to them. Like what
So
is the purpose of science, I guess, is maybe the point.
what is the purpose of science? I would say, being life sciences, is to feed into medicine and help treat disease and suffering. Pretty simple for a virologist to say that, but we rely on a huge amount of basic science, as some of your readers probably uh, would have difficulty understanding the arcaneness of some of our stuff. But there was, if you look at these phosphorescent blooms that you see in the evening on the seashore, there are some algae that have this phosphorescent protein. Well, this was cloned and developed into a wonderful tool for cell biology, and it was awarded a Nobel Prize. And this has been a phenomenal advance in biology, and in my field of virology, hugely important, hugely important. PCR, this polymerase chain reaction, the key point in that was not actually conceptual. The concept of amplifying by copying and copying and copying product, that was the first paper. The second was getting this enzyme from a bacteria that grows near thermal vents. You can just change the temperature, you can separate the DNA strands, the enzyme that does it does not degrade, and then you can start copying it. So in just a simple reaction, you go through this and you have this wonderful technique, PCR, which has absolutely transformed biology since what, for 30 years. Unbelievable. Nobel Prize in Chemistry, Carrie Mullis, a phenomenal. And from someone looking at bacteria in thermal vents, which you can say, but the ramifications, and there are so many examples of this. So we as virologists feed on all these things. We need this stuff. We need this science. If you just say to the virologist, just give me your life-saving work and we'll can the rest. I won't finance the stuff that doesn't give me a new drug, vaccine, or whatever. And I will have to say to him, you need that 9/10, that 90% to get that important 10%. Well, it's actually maybe 95% to get that important 5%. This is the way it works. But I think we don't pay enough attention to what we're doing to make sure that we are pretty sure that the way we handle microbes is close. I think we're handling in the lab in ways that are particularly irrelevant. And I think this is something that has to be explained and changed. Can I give one example? I don't mean to speak for too long.
Of course. Of course.
The body temperature of birds
Mhm.
is about 41 degrees. For us, that's fever, and we would die if that temperature is maintained. So you can imagine, if you're interested in a bird virus, you should probably cultivate it at 41 degrees. People don't do it. They use 37, which is our body temperature. Do you know the body temperature of a cow and a mouse isn't the same as humans? So if you're wanting, in thinking about mouse to a spillover of, say, of a Lassa virus from mouse to humans, well, maybe you should look at when you grow the Lassa virus in mouse cells, maybe you should choose the body temperature. People don't do this. There's so many more examples. I have an essay on it. Lab-adapted viruses and these little differences. And we can make an effort and people don't because it's easy. So I think there are lots of things that could be done, making things more physiological. The goal is not your damn career. It's to beat the hell out of microbes. Okay, that's it.
Should be. But how do we get there?
I would think that we get there by talking and discussing openly. It's not for me to tell people what to do. It's not for you to tell people what to do. It's for us to discuss and see, because there's clever, perceptive people out there. And together we could hone in on something and sort of say, yes, well, let's see if we can cultivate chicken flu viruses in a more natural setting. That would be a wonderful goal. The NIH could set that as a goal. Same thing for bacteria, and so on and so on and so on. But rather than me telling people, do this, do that, That's not the scientific way. We discuss and see if we can find, say, 10 ways to improve work on non-human viruses that are a menace to humans. Yeah, and you in your field in bacteriology, same thing. And just let people discuss rather than let the field develop it, rather than have someone say, "This is it."
So, Simon, I have a lot of ideas about this, and I also think the idea, like, it is important to have conversations, right? Like, that's the goal. But as we began, the discussion today talked about this Cell paper, and this is the problem. Like, this is not the fact that paper is is a is a high impact journal publication means that we're far away from having conversations in a way, a meaningful way. So it's great to say, how do we get there? You're right. We need to have conversations. I
Well,
think
look,
one of the reasons.
sorry, my apologies.
Well, no, I was going to say, I think a big part of the problem is scientists are trying to sell stuff. They're trying to market their stuff. They're not. We're scientists. We're not market managers. We're not PR people. We are scientists. You give results. That's it. That's it. I'm very convinced now that, like, a path to getting this ship righted is for us to just stop selling stuff. We don't need to sell.
So I
We
mean,
need to give results.
right.
Let the policymakers do what they will with our results. Don't sell it.
The policymakers are there to make sure that they fund the most creative and interesting science. The policy people are there to, when you have a big revolution like genomics or the cloning revolution of the late 70s, they were there to make sure that the infrastructure and particularly in genomics, and their grants were to develop computer programs to help assemble genomes. Hugely important. That was crucial. But they weren't to sort of tell us where the field should go. And they couldn't have dreamed of how cancer genomics has gone and advanced and is helping us understand cancer, and so on and so forth. They are there to help us with the technology and to get the money out to what they think to be the most interesting, but they shouldn't be telling us 2 and 2 equals 5 or 2 and 2 equals 6. It's for the scientists, the people doing the work, the recipients of the grants, who, through consensus, and of course some people speak too loudly and they brook little opposition, but discussion and unquestioning is a part of science, and this is.
But calling out bullshit, Simon, calling out bullshit, and this Cell paper is the bullshit. It's hype.
Well,
It's like, this is not the claims are over. They're overstating things. It's essentially just sells. It's BS. It's just not science.
that's right. I will have an essay coming out. It's called Trust in Science. And I fairly bluntly explain where science administrators can go wrong. And there we are. And I take the past NIH director and the present NIH director and make a comparison. You've got to be allowed to say you're wrong. You've got to recognize, freedom of speech. But you don't have the freedom to ram your ideas down the throat of someone else. There is no amendment saying you're allowed to do that. You're allowed to speak, but you can't ram it down someone's throat. And anyone who starts doing this is going to make science less efficient, because it's polarizing science, and politicians will be wasting their money because we're in a field of inquiry and discussion. And some outsiders do brilliant work. And then people say, gosh, how did I miss that? How did I miss that? The way hepatitis B grows. We go back now to the 1980s. There were some really strong people who've been working on hepatitis B for years and years and years. And there was one group by Jesse Summers, and I can't remember the name of the other person. Oh, goodness me. Anyway, Jesse Summers, an incredibly nice, in Philadelphia, a very nice, soft-spoken, careful, thoughtful person. And he worked out how the virus replicated. Now there's some big people, sort of bruisers in the field, And Jesse Summers got there because he was thoughtful. And he moved the field on. He moved the field on. Jesse Summers, became a classic in hepatitis B, and it helped us understand how you can make drugs, the dideoxy molecules that are being used for HBV. But so many people, you feel that he could have got it. Why did they not think about it? And Jesse, being this thoughtful, careful, soft-spoken person, just let his brain turn over, and he got there. Lovely story.
I mean, that's the way it should work, right?
Creative types. This guy, okay, with Paul, nearly 300 completed songs. What a track record! That's track record. That is class.
Simon, thanks once again for joining me on another episode of In Defense of Virology. This has been Bryce Nichols. Take care, everybody.
My pleasure once again, Bryce. Take care.